Well, I'm a bit of an old dog. I've been doing medchem for sort of 20, 25 years now. And one of the things, whenever you go to a medchem conference, what happens is that the sort of medicinal chemist gets up, stands in front of the screen, he says, normally puts up a picture of some horrible disabling disease or some crippling horrible graphic, and then says, well, this is the disease, this is the hypothesis, this is the target, and then this is our way to, to hit this target. And generally, more often than not, when you go to the conferences, it's actually they're not successful. And the point really being is that if they were successful, they wouldn't actually be talking at the conference. So that's the situation. Anyway, what I thought I'd do, I thought I'd try something different, and I'd talk about happiness. First thing I did was to go to the web, look up happiness. What's the graphic that I can include? Came across this one. It's the very nice, the chemistry of life. And what it says that if you're a schizophrenic, you've got too much dopamine. If you're either anxious or depressed, you've got too little dopamine. And if you're happy, it's just essentially a function of your serotonin level, which is why people eat loads of chocolate, make them happy. Of course, being the web, I then went to the next link, came up with this graphic, which essentially says, hang on, alarm bells immediately read, happiness is a function of dopamine. So, does this mean that sort of schizophrenics are just incredibly happy people? I don't know. I don't think so. But anyway, so I then went to the next graphic, which is meet your happy chemicals, and this is really essentially saying, well, it's a cocktail again. So we've got some dopamine, we've got some endorphin, we've got some oxytocin, and we've got some serotonin. So all these. What it also says to me, yes, we've got this cocktail, but it's actually we don't really know what's going on. We're a bit unclear. So that's current state of drug discovery, state of the art. The next slide, we've got a listing of typical and atypical antipsychotics on the bottom here. What it's also got is, the graphic really just represents that the molecules have a polypharmacology. So when you look at each of these, there isn't a single molecule there that doesn't bind to more than one receptor. So in particular, something like D2, so orange is a strong binder, D2 at the adrenergics, and the histamine, or H1 there. So what I'm going to do for this talk, or the story I'm going to tell you, or ask you to sit through really, is how do we go about designing a novel antipsychotic? Okay? Oh, sorry, I keep getting this wrong. Now, you've actually seen this graphic several times, I think, today. It's a sort of multi-objective optimization. I first saw this, I think it was by Darren Green from GSK actually presented this quite a few years ago. To me, medicinal chemistry is about, here's a graphic around, oh, I'm sorry. Ooh, giving the game away. Here's a graphic about, a typical fragment hit and the types of things that chemists would tend to do with it. So medicinal chemistry, several other people have said it before, focused on it. It's really about what to make next. So James Black said, a very good starting point for drug discovery was sort of natural hormones or natural targets. Here we've got dopamine. Kebble, very nice interface for putting out information, but what it really doesn't tell you sort of the relationships between molecules and targets. So one of the things that we did, and George was involved, or George Papadakis, who speak later, was involved in this, was a tool that we wrote called Kemble Space, where the edges, so the whole of the Kemble polypharmacology, so it's where the edges are proteins and the nodes essentially say that there's a molecule that hits more than one of those targets or those proteins. So how can we start? Well, we can say, so if I said I wanted my polypharmacology, my antipsychotic to be to modulate dopamine, modulate alpha-1 adrenergic, modulate H1, I can simply do something like search for dopamine in here in the text box, identifies all the dopamine genes. Similarly, I could search for adrenergic, pulls in the adrenergics, and then the edges here, there's a relationship between the two, together with their associated polypharmacology partners. Okay. I can also search for off-targets, or anti-targets, as it were, sorry, not off-targets. So histamine here, I don't want histamine. What's nice about this network view is the sort of this inference aspect, essentially, that okay, yes, I can see that there's a relationship, that I have molecules that exist that have both dopamine, H1, and adrenergic activity. But is there a relationship, is there any inference between, say, the adrenergic and, say, some of these molecules up here? So in other words, I know this molecule up here hits dopamine, I know it hits histamine. Is there something about this receptor that also suggests it's going to be alpha and adrenergic? You don't really get that when you look at graphs and tables and spots. It's this inference approach. And I think this is something I want to point out for some of the later slides. So I've got my molecule, I've gone to Chembl, I've pulled down all my polypharmacology data. What do I do next? Well, one of the things I might do, I might go and make some predictions around that molecule, further polypharmacology. So this is the C approach, the Kaiser and Shokat approach. And essentially what they're saying is I've got a molecule, which is, or my query molecule, and it's similar to this molecule, and I know this molecule hits this target, this target, this target, and therefore I'm likely, the inference is that I'm likely to hit my query molecule again. So that's the type of graphs that you can build. Now, this is great, and it works. We've had some good success with it, but the problem with this is that generally you get a high degree of false positives. You can well imagine as your molecules get larger, the sort of the descriptors that you use essentially become associated with other protein targets. So as I said, you tend to get lots of false positives. The approach that we used, we've developed, plenty of other people have done this as well, but we've developed in collaboration with Andreas Bender's group in Cambridge. It's, again, using Chembl data in this example. The phenotype was sleep. What we're doing is we're taking each of our targets or our structures, we're taking each of the compounds in Chembl, the targets, building fingerprints, building bioactivity profiles for those molecules. And then we're using a decision tree to actually fit the profile, the bioprints to the sleep phenotype. So we're using a naive Bayes here, and it's reasonably accurate. So the type of thing that we can do is, the tree looks like that. So the type of result that we get is, so for example, if we hit dopamine D2, if we hit H1, we hit 5-HT, then we get good sleep. So it's really nice and informative for the chemists. So we're using Chembl, or we use Chembl in this example, but we can also bring in the PubChem data, which is, in total now it's around 200 million data points. We use a combination of naive Bayes or support vector machines to do this predictive pharmacology. Where we are with this, we've got a very nice publication that demonstrated we took 18 drug molecules and tried to identify whether those, or predict whether those molecules would have some associated sleep effect. And the problem is that we can't actually get it published. We keep getting rejected. It's probably people don't want to see molecules that actually have an off-target effect. So we've got our molecules, we've got our known activity, our literature activity, we've got our predicted activity. What we're going to do next? Well, it's around how we go out changing the molecule. So we're all aware of this paper from Jean-Louis Monde, which is essentially the virtual enumeration of 17 heavy atoms, created 166 billion molecules. And the graphic really just sort of highlights how small our current compound collection spaces currently are. What's also not routinely reported that for something like, say, a typical pharma screening collection, which is normally sort of 100 to 500,000 molecules, is just about sort of 10 to the minus 5, 10 to the minus 6 % of that accessible space. So chemical space is absolutely massive. So how do we go about trying to design molecules, take them forward within that huge space? So the approach that people use, are things like is de novo design, or I've got a graphic here which essentially summarizes the strategies that people use in de novo design. So where you can take a molecule and you've got a core and you can essentially grow off it. You can take the molecule and you can replace the scaffold, or you can take multiple molecules in 3D and you might merge the cores together to swap functionality. This is great. The types of constraints that people use to limit this chemical space design are things like protein cavities, volume-based, which are ligand summations, or feature-based. And the main issue around de novo design, or seems to be around synthetic feasibility. Can I really make those molecules? And there are several strategies that people have used to try to circumvent this problem. Our motivation in terms of de novo design was essentially with the sort of introduction of ELNs and lab notebooks. So what we have, or I was working at Lilly at the time, we had something like 500 chemists. You know, if they're doing 10 reactions a week, that's 250,000 novel reactions a year. Now, we really ought to be using that data for future design. It's a huge amount of reaction data. The approach that we developed was around reaction vectors. It was actually inspired from work by some of the Merck guys in 2003, where they're looking at reaction similarity. So a reaction vector takes the reagents, takes the product, and actually subtracts the descriptor of the reagents from the product vector to give it's a difference vector. In terms of structure generation, what you can do is actually switch it around. You can say, well, actually, given a reaction vector D or transform and a set of reagents, you can actually generate a new product. Does it work? Well, given those simple descriptors there, we can generate those multiple or 3 different products. So what we've done, or the conclusion there was really we need a better descriptor. The descriptor that we came up with, this was work done by Hina Patel for her PhD thesis, was these atom pair descriptors. We tried a whole variety of different descriptors and different fingerprints, but these are the ones which we felt worked best, were simple enough. So they include the element, the number of bonds to heavy atoms, number of pi bonds, number of ring membership, and the bond order. We actually work with atom AP2s and AP3s, which are, if you like, the extension or the one bond extension from AP2s. So how does it work? Or what do atom pairs look like? Or what does the reaction vector look like? So here's a simple Beckmann rearrangement. Okay, so oxime to acetamide. So you have a series of negative atom pairs, and we have a series of positive atom pairs. So the negative atom pairs are the atom pairs that were actually removed during the reaction. So here's an example. So here's my oxime, acetamide. I remove my negative atom pairs, and I get these functional, this collection of fragments. Next step is to apply the positive atom pairs to the fragments. So I apply it's essentially building Lego. Or I apply my positive atom pair, my fragment, to my starting fragment collection. I go through in each turn, and actually then I apply my AP2 and just check whether that AP2 fits using my AP3. How well does it work? Well, in the original implementation, we had something like, reasonably well. We had about 90%. This was done, work by Dimitar Christosov. It was a collection of a variety of different known organic reactions. Essentially, we got 90% of them right. And this is actually an organic chemistry database. So this isn't your normal traditional medchem. This is making horrible sort of complex macrocycles and things like that. The type of places where it didn't work are things like Diels order reactions or Claisen condensations, these sort of horrible rearrangements. Where we've gone now, what Dim went on to do was to actually rewrite the algorithm and include the sort of bond path and the fragmentation process. We can get this up to sort of 99.6% at the moment. So how are we using it? Well, what you can do, you can take a fragment. You can take a fragment, you can apply a reaction transform from the reaction transformation database. So to generate a pool of possible products, you can score those products either via local models for QSARs or docking scores or global models, things like ADMET, QSARs or pharmacophores or Similarity or whatever. And then you can take off your top population or cream off your top population, go back round and remutate to generate a new set of products. And you can go around several times or as many times as you want to do the optimization. This is I've got multiple examples. This is a simple example. So I've got my 2 antipsychotics, typical and atypical. I'm going to say, so my score is based upon, say, the union of the description of the pharmacophore fingerprints. So I'm designing molecules that are similar to both of these. I want to have a good D2 activity based upon a simple QSAR. a good alpha-1b adrenergic activity, and a loss of or limited H1 activity. That's a typical score. So here's an example. I was using 23,000 reactions, 93,000 reagents, start from the pipridone, and end up with this Spiro tricyclic system. So, perhaps this is a noddy example, and yeah, I've got some. We've actually found some molecules. These are known antipsychotics, and yes, they look very similar, but probably they're included. I didn't check; they were probably included in the QSAR. So it's we're driving. The QSAR is a similarity based, so we're driving to those molecules. What's nice about it as well is that we get the reaction scheme. So we get the pipridone, the alkylation, the second alkylation, the spirocyclic formation. And here the chlorination. You probably don't want to do it in this route, but at least it gives you an idea. Okay, it looks great. So what we've developed is essentially a way to take a molecule, have a reaction database, and automatically go through what can I actually do to that molecule? What new molecules can I build? It looks great, but the algorithm only knows about the transformations that are in the database. But that's both its strength and its weakness. The point was we were going to take a known reaction data and be able to apply that. That's our feasibility aspect. Second point is the AP2/3s cover only the one or two bonds, and so remote functionality isn't considered. And you may think that's a problem, but it's actually not a huge problem. I think that the problem that we struggled on was that actually a reaction path is not optimization. So you're trying to do this multi-objective optimization, and when you look at this example, this was work. I've forgotten his surname now, but a guy called Ben did a postdoc on it, and he spent 2 years trying to do a multi-objective optimization, and we essentially came to the conclusion we couldn't really do it. So we got a lactone, we open up the lactone, we protect it with this TBDPH functional group, silyl group, tetrazole formation, and then deprojection. And the point is, if you're doing a multi-objective optimization, for something like this, if you've got a molecular weight cutoff, then you're really going to very readily eliminate that molecule. And yet that's probably the best route into that molecule. So the question really was how do we get from here, A, to E? So the way that we've got past this is the introduction approach we've called reaction sequence vectors. Okay? And it's because we're using these difference vectors, because we're not doing chemistry as such, we can encode from A to B, we can code from A to C, A to D, A to E. The computer doesn't care. We can go from B to C, B to D, B to E, and so on. So we get these networks where the molecules are nodes, and the RVs are, the transformations are essentially the edges. We can also do it the other way, where the RVs are the nodes and the molecules are edges, where you really learn about the chemistry. But in terms of de novo design, this is the approach you want to focus on. Now, this is work been done by a guy called James for his PhD. He's just about to publish how sequence vectors are a better approach on de novo design. But I'm not going to go into that because will preempt that publication. For his thesis as well, he also presented this, which is how you can use these sequence networks for SAR exploration. So here's our starting material. This is a typical medchem paper. Okay, from the acid, again to the oxime. And what you would see reported in the paper would be something like this: a series of SAR results, whether one or two, a target plus an anti-target. So we can use our sequence vectors. So we can take our starting material, we know the route, and we can say along the route, well, what can I do to that? And I can produce these networks. And I can also, in this example, which is probably one of the most simple examples we've also got novel routes into that pathway that we can identify. Okay, that's great. So what does that mean? So what I've got here is I've got a PCA, principal component analysis of the property space. So we've described our molecules, we've done a principal component analysis of the properties of those molecules. The known products are in blue, those ones in the paper, but the ones in red are actually the near neighbors, okay? So when we, quite often if we have a hit, we'd probably go off to something that's quite close and quite similar, just to make a small change to a molecule. But what we can do in terms of using the sequence vector approach is we can create a much larger property space. And then, so the way that we're going forward with this is we're currently developing an approach around active learning to select the best training set. This is a mapping of discovery space. So this came from 600,000 reactions from Daniel Lowe and the Next Move guys. So where each of these molecules is a node and the edge is a sequence vector transform. It's large. One of the things that, when I sat down and thought about this, how can we use it, is really we can think about, well, what happens when we, essentially we've mapped out feasible chemistry space for a molecule. So we can take our molecule and we can say, so here's our active, here's our hit, where can we go with it? If our blockbuster is here, I'm sorry, I've just hit the wrong button here. So if our blockbuster is here, if our blockbuster is here, our hit is here, what you generally do with sort of a His assessment would be these very simple transforms around this active molecule. But if we map out the feasible chemistry space that we can apply, well, we could think about, well, actually, maybe drug discovery isn't necessarily an optimization anymore. Maybe it's actually just a sampling problem that we need to think about. So we can go sample even further, more rigorously. One of the things that you can do with this discovery space, if you go back to the original algorithm, the difference vector, it's very similar to sort of matched pair approach, where you take one molecule and you look at the differences relative to another molecule, and you can map out onto the reaction network your matched pairs or even simple QSAR activities. Okay, I'm going to change gear. I'm going to talk about other network approaches. This is some work that did at Lilia, which is around the genome-wide psychiatric genomics consortium. So this is here we have a Manhattan plot for schizophrenia. This is chromosome 6. There's a very faint line here. Anything above this line essentially says from epidemiological studies that if there's a mutation, it's probably possibly associated with schizophrenia. What interests me about this Manhattan plot is thinking about, hey, there's multiple mutations that are associated with schizophrenia 1, but there are also multiple mutations that are actually in non-coding regions. So thinking about, well, actually, God, I've got to design molecules that actually hit both coding and non-coding regions. How do we do it? It's a difficult problem. The way that we targeted it, so in red, I've got this was some work that was done by Suzanne Brewerton, who's moved on from Lilly now. We took the schizophrenia GWAS genes, we knew the GPCR receptor subtypes, and we did a shortest path analysis to identify the targets or the genes that were associated with those GPCR receptor subtypes. So we didn't do it for 3, we did it for multiple GPCR receptors, and you can come up with a sort of an aggregate that looks something like this. If you remove the proteins that were the degrees of less than 10, you can identify the hubs, and these are the hubs that are involved in GPCR signaling in schizophrenia. And you can see where we are. That I've already talked about the multi-objective polypharmacology design. So we can use tools like that to actually make some suggestions for molecules that target those hubs. The other approach that we've taken, again, this is work that we've been doing in collaboration with the Bender Group, is to work in the gene space. So we have our geo database, where we've got our collection of our microarray and seq-RNA data. So of disease phenotypes. So we know the gene expression of our disease. There's a database called the Connectivity Map, which is held at the Broad Institute. And this is a collection of gene expression. So when you add a compound, what's the gene expression that's resulting in that addition of that compound? There's 2,000 compounds in the CMAP collection. There's 20,000 in the links collection. And what you simply do is you look for complementarity between the disease gene expression and the compound gene expression. What we did with the Bender Group, at least, is just to add in the sort of polypharmacology prediction, and also use the sort of a network pathway approach to try to really pin down what's the most appropriate compounds to take further. That's network discovery, looking in the whole cell, looking in the gene expression level. But we can also do, and this is quite common now, I think we've already seen some workflows, or the drug discovery workflow, but I think the drug discovery workflow, there's a sort of a new paradigm shift as people move to sort of or with the advent of sort of more high-throughput image analysis, people have shifted more to sort of a phenotarget or phenotypic approach and target deconvolution. And this is the workflow that we use at Evotec. So we can take our compound selection, we do a phenotypic screen, we build our SAR, hit to lead, and identify the target. So what's different here is that so we can when we put our compound collections in the same way that Terry talked about, we can focus on chemical diversity, but what we now can do is also think about the biological diversity and the annotations around those compounds. I mean, so what's the point in just looking at structural diversity if I know that several of those compounds will probably hit the same target? You want to really, at the end of the day, we want to hit a biological target. We can take our structurally diverse hits, but we can also take those hits and we can look at the pathways that are associated with those hits, and then we can explore them with things like predictive pharmacology. We can do transcriptomics approach. We can do things like cellular target profiling and proteomics. We can do knockdown studies, things like CRISPR technologies, where we actually mutate specific genes to either knock down targets to inform us of our appropriate target. So here's a typical workflow. I think where I am, or the point I'm trying to make is that we no longer should be working in this sort of cheminformatics space, but we really need to integrate the target data, the bioinformatics data, to really bring things to bear. So in this example, I've got Chembl, PubChem data. I can use the predictive models. I can annotate. We've got a couple of collections, we use a 6K collection, which is very highly annotated of drug molecules, but we've got a 20K collection as well. Take our annotated collection, we do a phenotypic screen, we do a gene enrichment, we can enrich our targets, we can enrich our pathways. What's the next step? Well, the next step is then thinking about what other database information can we apply, can we add into that process. So we can use things like OMIN databases, knockout mutation databases, clinical data, things like toxic effects from cider. We can then take our target list, we can search for compounds which are particular target pharmacological probes, and we can use this literature evidence. So this is a huge amount of data. What we're trying to do is we have this hypothesis, which could be considered to be a big map, and we're really focusing down to sort of identify where our particular town or where our target is based upon the huge data. What's interesting about this is, or the bottleneck currently is around, well, actually this all sort of needs to be done by the guys that are really expert in the disease. There's a quick example here where we've done a phenotypic screen. We've identified from the compounds, the targets, exact match. If we use similarity to those compounds, we can identify more targets. If we use predictive molecules, we can then identify even more targets. The issue around that is, of course, well, actually I've gone from 146 targets that my biologist needs to sift through to now to something like 226. So the problem becomes even more complex. So it's a challenging problem. We have been successful. We've published in PLOS a guy called York Rudhard for target deconvolution. Okay, so for me the discovery challenge is what we can do, where we're at. We can now take a molecule and we can map out the chemistry space around that molecule. The goal really is to relate this chemistry, for me, is given a disease signature, how do we best sample the appropriate chemistry space? So it's this relationship between gene data and chemistry data. So sadly, I haven't discovered a new drug to treat happiness. What I found is this quote here, and this is a second quote around happiness that was in the meeting, and we're also with Stephen making chemists happy as well. I think the theme has somehow revolved around happiness. But that's the talk. I have one last slide, which is the acknowledgments. A lot of the reaction vector stuff that was done with my key collaborator over several years and several postdocs, PhDs, Val Gillett. My group at Lilly, including George, some of the other guys, Nick Fechner, and my collaborator at Cambridge, Andreas Bender, the guys at CCD who've funded the multi-objective postdoc, and my current group, or some of the guys at Evotec at least. So thank you.